Protein synthesis is the process in which protein is produced with the information from the DNA. This process occurs in all cells, but in this model, the cell is a eukaryotic animal cell. As we zoom up to the nucleus, you can see that the nucleus has nuclear pores which act as openings for the RNA to enter and exit the nucleus. Inside the nucleus is DNA, which holds all the information. The DNA information is put by the different combinations of the nucleotides. There are 4 types: adenine, thymine, cytosine, and guanine. There are paired nucleotides that match up with one another. For example, cytosine and guanine always goes together. You can think of Cinderella and the glass slipper to help you remember. The other pair is with adenine. In DNA, adenine pairs with thymine, which can be remembered by Ariel and Triton. But in RNA, adenine goes with uracil, which can be remembered by Ariel and Ursula. Transcription is the first step of protein synthesis where the mRNA copies the DNA gene. First, the RNA polymerase, an enzyme, unzips the DNA, which is usually in a double helix shape. Then, the mRNA or messenger RNA that came through the nuclear pore copies the code from the DNA by matching up the nucleotide pairs, one base of the pair being on the DNA template strand, the strand that gets copied, while the other is on the RNA. The TALTA box, or thymine, adenine, and so on, is a promoter on the DNA that lets the RNA polymerase know where to bind. RNA processing comes right after transcription, which ends when the RNA reaches the termination signal or a sequence of specific bases that signals to stop. After transcription, pre-RNA is made, and this is turned to mature RNA after RNA processing. In RNA processing, the introns or non-functional segments of the DNA are taken out while the exons or expressed DNA are put together. The results create what is known as mature RNA. After RNA processing, the mRNA goes out through the nuclear pores once again to a ribosome. Ribosomes are the site in which the proteins are produced. They have a large and small subunit. At the ribosome, the process called translation occurs, and it has 3 steps: initiation, elongation, and termination. During initiation, the start codon, or AUG, attaches to the small ribosomal subunit, and the large ribosomal subunit attaches to the smaller one, sandwiching the mRNA in between. A codon is a combination of 3 nucleotides that make an amino acid. The opposite of the codon that matches it is the anticodon, which has the opposite of the basis of the nucleotide pair that is on the codon. Elongation, the second step of translation, is when tRNA, which is clover leaf shaped and holds the anticodon, moves into the side of the ribosome, 2 at a time, to attach the codons and anticodons. Peptide bonds are formed between the amino acids. The last step of translation is termination. In this step, the final product is produced, which is the primary structure of a protein, which is basically a sequence of amino acids that are bonded together by peptide bonds. And that's the end. Goodbye!